Hello students, welcome to the EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am senior scientist in the division of animal biochemistry at National Dairy Research Institute, that is NDRI, Karnal, in Haryana, India. I am going to talk about the overview of bioinformatics from the paper Biostatistics and Bioinformatics. Bioinformatics began with efforts to combine experimental biological data with computational algorithms for generation of information and knowledge about the biological processes. The ultimate goal is to model complete biological processes which are recognized series of molecular events or functions. Each biological process has a defined starting function and a defined ending function with intermediate functions as in biochemical pathways. These functions or the activities are essential for integrated functioning of the living cells. Bioinformatics began on a small scale with the development of sequence alignment algorithms for searching homologous sequences which are conserved during the evolution. The power of sequence alignment for searching similar sequences from the database was appreciated with the computational gene hunting for the disease cystic fibrosis. Subsequently, bioinformatics encompassed high throughput technologies developed using biochemistry, molecular biology, biotechnology, genetics, chemical engineering, mathematics, computer science, information technology, and telecommunication tools. These new foundations were used for the collection of the biological data, organization of biological data, mining of the gene and protein, that is biological data, including even the three-dimensional structures and biochemical functions, that is pathways, biochemical pathways, with the ultimate aim of modeling biological processes of living and functioning cells. Let us now have a look at the learning objectives in this module. We'll begin with bioinformatics in relation to biochemistry and biotechnology. And this will start by taking an example of bioinformatics, that is the classical example of cystic fibrosis disease. Then we'll follow to understand the central paradigm of bioinformatics. As there is central dogma of molecular biology, there is central paradigm of bioinformatics. And we'll continue with defining what are the key areas in the area of bioinformatics and how the bioinformatics originated. It emerged as the fusion of biotechnology and information technology, and then we'll continue to understand what is the aim and goal of bioinformatics and what is the scope of bioinformatics. Now we'll discuss gene hunting of cystic fibrosis gene. The disease cystic fibrosis is associated with abnormal secretions leading to recurrent infections of respiratory tract. Cystic fibrosis was diagnosed in 1 out of every 2,500 persons in Caucasian race of Western population. 1 Caucasian out of 25 is a carrier of the faulty gene present on one of the 23 pairs of chromosomes in humans. Its gene is autosomal and recessive. By autosomal genes, we mean that the disease is not sex-linked and the gene is present on a chromosome other than sex chromosome X or Y. This means that the gene is present on any of the other 22 pairs of the human chromosomes. In addition, the cystic fibrosis disease is a recessive trait, which means that when both of the alleles on each of the homologous chromosomes are faulty, only then the disease is manifested. Now, with the advancement of laboratory testing, the presence of cystic fibrosis disease is well documented even in other countries, including India. Interestingly, the most of the phenotype of cystic fibrosis gene is in accordance of the Western population. In 1980, biologists knew nothing about this gene. The search of the cystic fibrosis gene began in early 1980s with genetic mapping, using recombination frequency of genes and RFLP probes as markers. In 1985, 3 groups of scientists independently announced the cystic fibrosis gene on chromosome number 7. The gene was located between MET gene, a gene involved in cancer, and D7S8, an RFLP marker. The genetic map encompassed approximately 1 million nucleotides. Genetic mapping was followed by physical mapping. This began with generation of complete genomic library of overlapping clones generated using different restriction enzymes. A clone hybridizing with D7S8 marker was selected for next stage, that is sequencing. The physical mapping was followed with actual nucleotide sequencing of the clone hybridizing with RFLP marker D7S8. This nucleotide sequence was used to design a probe for selection of next overlapping clones hybridizing with the ends of this sequenced clone. After several rounds of selection of overlapping clones and their sequencing, the gene of interest was ultimately sequenced. Knowing the approximate location of the gene, however, does not lead to the gene itself. For example, the Huntington's disease gene was mapped in 1983, that is well before cystic fibrosis gene in 1985, but it remained elusive till 1993. However, with the overnight database search in 1989, a region encompassing about 6,500 nucleotides was found to have sequence similarity to ATP binding proteins. This sequence database search not only identified the gene but also hinted at its possible structure and its possible mechanism through which the disease was manifesting. This showed the power of sequence similarity, which was based on sequence alignment for searching similar sequences in the databases. This sequence similarity search is most widely used tool of bioinformatics today. This laid the foundation of bioinformatics. This database search revealed that the protein encoded by the cystic fibrosis gene is a transmembrane conductance regulator, which is an integral membrane polypeptide of 1480 amino acids. This polypeptide has 5 domains. These include 2 transmembrane, 2 cytoplasmic, and 1 regulatory domain. Each domain has a specific function involving transport of chloride ion from environment or from inside to the cell through the cell membrane. Consequently, mutations in these domains affect the transport of chloride ions through the cell membranes, causing abnormal secretions in the respiratory tract or even in the sweat. You will find that when the cystic fibrosis is there, the salt comes and manifests the disease. The most common symptoms are the sweat and respiratory tract infections. we are well aware that biology at the molecular level is guided by central dogma of molecular biology. Central dogma of molecular biology tells about the flow of information from DNA to RNA through transcription, and then from RNA to protein through translation. And this flow of information is never in the reverse direction. That is never back from protein to RNA or from protein to DNA. This shows that the central dogma of molecular biology dictates the direction of synthesis of biological molecules. That is from DNA to RNA, from RNA to proteins. This dogma forbids the synthesis of nucleic acids from proteins. Never happens. This flow of information then decides the manifestation of the phenotype in a given cell or an organism. However, this dogma is not able to answer the questions like that when a gene in the life cycle of an organism or a cell, or in which tissue of an organism, or why a gene is expressed at all, or how the phenotype of the gene is manifested, the mechanism in an individual. Central dogma of molecular biology simply conveys that the sequence information determines the phenotype of a living organism or cell. The central paradigm of bioinformatics, on the other hand, considers that sequence information in DNA, RNA, and proteins represents the same knowledge and therefore determines the molecular structure of actual transforming machines, that is proteins and RNAs. These molecular structures transform metabolites and therefore controls the phenotype of the biological system. Therefore, central paradigm of bioinformatics specifies that the sequence information determines the protein structure, which in turn will give specific biochemical functions to finally express the cellular phenotype through interaction of metabolic, signaling, and gene expression pathways. The cells which will be surviving will then reproduce to pass on the genetic sequence information to the next generation of the cells, which will repeat the sequence to structure to function to systems cycle. Therefore, bioinformatics is having 4 sequential levels. The first is the determination of genetic sequence information encompassing genes and proteins. The next is the transformation of this sequence information into 3-dimensional structural information of the proteins and RNAs. The classical experiment of Christopher Enfinson in 1973 involving denaturation and renaturation of RNAs established that the information for folding a protein into 3-dimensional structure is encoded within the amino acid sequence of the protein itself. Therefore, once the protein is synthesized, it will fold into 3-dimensional structure as per information contained in the sequence itself. Once the protein folds into the encoded 3-dimensional structure, then this protein and other interacting structures implement the biochemical transformations, resulting in signal cascades and biochemical pathways. These pathways then interact with each other to express the phenotype of the cell, dictating systems biology. Those phenotypes which survive this cycle are able to produce or reproduce the next generation. Now we'll have a look at the key areas of the bioinformatics. Bioinformatics encompasses computational molecular biology tools, that is, mathematical and computational analysis of sequences and structures of biological macromolecules. Genomics encompasses application of computational molecular biology tools in a large-scale genome research targeting investigation of the complete set of genes and their RNA as well as protein products. On the basis of the central paradigm of bioinformatics, bioinformatics has 2 key areas. Structural bioinformatics and functional bioinformatics. Structural bioinformatics refers to the gene mapping and determination of DNA, RNA, protein sequences, and structures on a large scale. Functional informatics is concerned with the annotation of existing structural knowledge about DNA sequences and their diffusible products with functional knowledge. It has two successive levels: study of expression of complete genomes at RNA level, that is transcriptomics, and at protein level, proteomics. Consequently, there is a paradigm shift from static structural bioinformatics to dynamic functional bioinformatics, as well as modeling complete biological systems. Structural genomics at DNA level is based on meiotic recombination frequencies involving genetic maps, which store distances between genes and other markers. Based on nucleotide lengths, physical maps store distances between these genetic markers. At the ground level are the sequence maps, which store nucleotide and amino acid sequences of the genes as well as proteins. At protein level, structural bioinformatics elucidates the representative sets of protein folds and domains. This is followed by mining the databases for retrieval of useful information, leading to the knowledge discovery using sequence alignment, similarity search, computational gene discovery, phylogeny, comparative genomics, protein structure analysis for function assignment. Functional genomics is concerned with attaching functional information with the structural knowledge. At transcriptomics level, high throughput analysis of gene expression is carried out using RNA and DNA microarrays, serial analysis of gene expression, and expressed sequence tags. Similarly, proteomics is concerned with protein level measurements of gene expression using high throughput technology. The aim in proteomics is the quantitative measurements of translation products under normal and disease conditions to appreciate differential protein expression so as to discover novel protein targets for drug discovery. For functional genomics using transcriptomics, several technologies have been used. The most widely used technology is DNA microarray technology, in which the cells under normal conditions and experimental conditions are taken and their complete mRNAs are isolated. After isolation of the mRNA from both the types of cells, the mRNA preparations are labeled using fluorescent dyes. In the case of normal cells, the mRNA is labeled with a fluorescent dye emitting green color, and in case of experimental cells, the mRNAs are labeled with a fluorescent dye emitting red color. Green and red colors were chosen because at the next stage of the DNA microarray technologies, these two preparations are to be mixed in equal amounts. Once we mix the two colors, that is green and red, in equal amounts, we get a yellow color. Therefore, when we mix them in equal amounts, if the color is yellow, it means both the green and red color are present in two preparations in equal amounts. After mixing, both are hybridized with the DNA microarray chip and they are scanned. When they are hybridizing with the DNA microarray, the mRNA preparation, mRNA molecules in the preparation will hybridize with the specific DNA probes attached to the DNA chip, the DNA microarray chip. This chip is a matrix of all the probes arranged in rows and columns. Each cell in this chip represents a particular specific gene. The gene probe is attached on all the spots in rows and columns. Therefore, if we have a chip, let us say for 100 genes, and then we'll arrange the chip in such a way that there are 10 rows and 10 columns, and we'll have 100 cells. In each cell, we'll attach a specific DNA probe which will hybridize with a specific mRNA product of that gene. When we hybridize the preparation of equally mixed mRNA preparations, then the mRNA present, if in the preparation, will hybridize and bind to the DNA probe on the chip. If it is not present, it will not bind, and therefore there will not be any signal. In these cells where there is no binding, there will be no signal. The color of the spot will be black. Where the gene is expressed only in the normal cells, therefore the gene is not expressed in the experimental cells, the mRNA which is labeled with green probe will bind to the target probe, which will then emit only green color. Since the mRNA from experimental cell is missing, red color will be missing in this, and since red color is missing, the color will be pure green. Similarly, when the mRNA is present only in experimental cells, it will hybridize with the target probe on the chip, and that spot will look red in that case. In those cases where the gene is equally expressed both in normal cells and experimental cells, therefore the two colors will mix in equal amounts. They will bind equally to the target probe on the DNA microchip, and therefore they will give yellow color to the spot. Where in other cases we'll have 60-40 ratio, 40-60 ratio, we'll have a gradient of colors, and scanning machines can read all these gradients and transform this color into the quantitative levels of mRNA expression. Therefore, we'll come to know that where there is green color, the gene is expressed only in normal cells. Where there is there is red color, the gene is expressed only in experimental cells. Let us say the experimental cells are a disease condition. If red spots are there, this shows that these may be the mRNAs and consequently the proteins which are involved in the expression of the phenotype of the disease. Therefore, one may look for novel targets for designing drugs targeting these genes or their protein products. On the other hand, when a gene is expressed in normal cells, its color will be green, and if it is not, when it is not expressed in experimental cell, the disease may be because of the downregulation or blocking of the expression of this gene. Consequently, the green and the red signals, which are pure signals, provide us opportunities to select the targets for the drug design. Similarly, we can check the expression of genes at the protein levels. The technology involved in this case is two-dimensional gel electrophoresis. In this case, of two dimensions of the gel electrophoresis, the protein sample is first run into first direction in which STS page process occurs, and the separation of the proteins is based on their molecular weight. After the separation of the proteins on the basis of their molecular weight, the gel is then run into the second dimension based on the isoelectric focusing. Now, the proteins in each separated band will further get separated on the basis of their isoelectric points. After the separation of these proteins in the sample in two dimensions, we'll get several spots on the gel which can be scanned for quantification. through phosphor imaging, and they can be identified using mass spectroscopy. For identification, each spot which is there can be picked using robotics and then digested with specific protease cleavage enzymes and subjected to mass spectroscopy for sequence determination. This will then establish the identification of the protein based on its molecular weight and sequence. Therefore, we can now compare. First of all, we can resolve all the proteins from a given sample. Then we can take a diseased sample and then compare two gels for the up and down regulation of the protein. The spots which are present only in the disease condition or the spots which are present only in the normal condition can be compared and can be used for identification of the novel targets for drug design. There are two types of machines or systems. One is living, other is non-living. Both types of systems or machines, they transform matter, energy, or information. Biochemistry is the study of chemistry of life, and chemistry is the making and breaking of chemical bonds in the living systems. The living systems are organized hierarchically in the form of an individual component contained in a container, where the container may be component in higher level container. The largest living container on this earth is biosphere, containing various ecosystems, having various species in each ecosystem, and in each species containing various individuals, that is organisms. Each individual is then made up of various types of cells with several organelles contained in the cell, and the organelles ultimately made up of large number of macromolecules. Biochemistry and molecular biology is concerned with the collection and analysis and interpretation of data about living components under normal and abnormal situations, so that corrective steps or measure can be taken if any of the living component behaves in some undesirable manner. The approach followed has been reductionist, with isolation of single living components, say a protein or a DNA or a gene, and then making observations experimentally on this living component. Life is defined as the capability of a system to reproduce itself, as a cell can give rise to another cell. That is to produce a replica of the complete system. A DNA produces a complete DNA, a cell produces a complete cell by itself. However, this ability of living systems is made possible through making and breaking of chemical bonds at appreciable rates, that is very fast rates. Otherwise, the same chemical reactions are going on in the non-living world also. With bioinformatics, now there is a transformation from the basic reductionist approach of biochemistry, biological sciences, of observing single component like a protein, to a holistic synthetic approach to combine and organize all the components in a container on a very, very large scale, so that a container is generated or simulated or modeled. So the development of these models help in making predictions about complete living systems. So the ultimate goal of the bioinformatics is that the biological components, that is molecules, macromolecules, are organized into organelles which then form the cell. So the molecules or the macromolecules which are there in the living system, they belong to particular biological processes. For example, glycolysis has 10 enzymes and they connect various metabolites in a pathway, and this is complete biological process. The aim of bioinformatics is to model or simulate biological processes so that the predictions are made about them to regulate their functioning. Now we'll see the emergence of bioinformatics with merging various disciplines. Previously, various disciplines were carved out as and when sufficient specialization was achieved. From chemical sciences, studying inorganic world, biological and physical sciences diversified into various disciplines. Then the era of merger of disciplines began, with biotechnology emerging through merging of biological sciences involving biochemical engineering, genetics, and microbiology. This era was followed with information technology emerging through merging physical sciences involving electronics, computer science, and telecommunications. Subsequently, bioinformatics, born out of the alliance between existing disciplines of biotechnology and information technology. Tools and techniques for bioinformatics are derived from multidisciplinary combination of various disciplines involving biotechnology and information technology. Now we are able to define bioinformatics. There has been development of sophisticated high throughput technologies in biological sciences. These high throughput technologies are low cost and they are very fast to acquire large amounts of biological data. Therefore, high throughput technologies are able to acquire and analyze large quantities of gene sequences and structures as well as gene expression of complete genomes at a very high speed. The quantity of data produced is so large that it is impossible to manage this large-scale data without automation using computing and robotic devices. Consequently, mathematics, computer science, and information technologies were increasingly applied to collect, organize, and interpret this large quantity of data for modeling biological processes. Therefore, bioinformatics can be defined as a discipline for collection, organization, and mining of large quantity of biological data for modeling biological processes. Now let us have a look at what is the goal of bioinformatics. Bioinformatics has emerged as a result of advancement of the experimental technologies for collection of vast amount of data in biochemistry and molecular biology, and subsequent organization, interpretation, and modeling these data with mathematics using computers. Bioinformatics therefore fuses biotechnology with information technology, where biotechnology provides data for IT processing and IT makes prediction about biological processes to generate information which can then be tested for its correctness through on-field, on-bench experiments. Dear students, in this module, we have seen the emergence of bioinformatics after merging the established disciplines of biotechnology and information technology. And with the development of bioinformatics, we are able to achieve those aims and goals which were not possible. Suppose we are interested to take a snapshot from history of life that extends back to some 2.5 billion years. But we find no experimental technology except to have some information from DNA fossils. On the other hand, with bioinformatics, we can expect to model functional system, that is complete biological processes in that age. This is hopeful. Similarly, with bioinformatics, we can expect to predict that in future, there may be evolution of a deadly virus. and therefore design the strategies to combat that evolving deadly virus in advance. Imagine a scenario in future in which a new deadly virus creates a pandemic of some fatal disease. Laboratory scientists will provide the genetic material for automated sequencing, and computer programs will then take over to design an effective drug against this deadly virus. Comparative genomics of this new virus with previously studied viral genomes will suggest some targets for developing drugs against this new virus. Three-dimensional structure prediction of the target shall then provide the actual site for action of the drug. Say an inhibitor of an enzyme sits in the active site and inhibits the enzyme, and therefore disease is deleted. High throughput virtual screening, that is pharmacogenomics, may then lead faster drug development from the predicted drugs, which may be finally tested in the field for its efficacy. Therefore, the scope of bioinformatics is to model complete living biological processes so as to predict the behavior of the living systems and to take corrective action if any of the processes behaves in an undesirable manner. And all this for better health, nutrition, and environment for the living systems on this earth. I thank you all for visiting EPG Pathshala.